Okay, good afternoon. We will get started. I wanted to mention for those who have asked me, if you attend 50% of the sessions during the course of the year and you take the final exam, which is a multiple choice electronic exam that you can take several times, it's nothing you should lose sleep over. But if you pass that, or when you pass that, you'll get an electronic certificate to say that you took this course in demystifying medicine, and it may be of some, hopefully, of some value to you. At any rate, the final exam will be electronically available by the end of the month, hopefully a little bit earlier. I'm trying to write an exam that I can pass. So today's subject of drug resistance in cancer, certainly needs no introduction. It's a beautiful example of the sort of theme of this whole course of the Brooklyn Bridge and connecting what was two different disciplines. And in this case, of course, the disciplines of clinical oncology and the big problems of cancer biology and pathobiology. Where is... okay. So our two speakers today are Michael Gottesman and Mauricio Barato. So let me tell you a little bit briefly about each of them, but I suspect that many of you know a great deal about them. I was kind of thinking on walking over here that it was almost 30 years ago. Michael and I had dinner in Holland after a meeting. I was at the Albert Einstein College of Medicine. I didn't know much about the NIH, except I admired it a great deal. And he told me that he had just been appointed as the deputy director for intramural research. And I must confess, I didn't have a clue as to what all that meant, but it sounded very important, and I liked Michael and congratulated him and so forth and so on. It took a long time. In fact, it was only until about maybe 10 years ago when we wound up here that I began to realize that this is not a job. This is the job. This is one of the most difficult jobs that one I would visualize could ever hope to be because, it blends skill, knowledge, patience, and an enormous number of other attributes. And Michael has been the deputy director for intramural research since 1976. And we are all deeply indebted to him for all of his efforts. Now, as a physician scientist, Michael graduated from Harvard, took his medical training there, trained in molecular biology here with Dr. Gellert, went back to Harvard, came back to NIH, and has been here ever since. But Michael is the chief of the laboratory of cell biology in the NCI, and that has been one of the certainly preeminent laboratories in the world for studying drug resistance, and particularly in cancer, and understanding more and more about mechanisms and development of ways to get around this problem, which in cancer is perhaps the inevitable and fatal step in the disease. So I was thinking about Mauricio on walking over here, and you'll permit me for telling another brief story. But a long time ago, a famous conductor, Bruno Walter, was due to conduct the New York Philharmonic, and everyone came to hear him, but he was sick with the flu. And a young protege named Leonard Bernstein took the podium for the very first time. people began to think about Volter afterwards, but not with the same gusto, because Leonard Bernstein just swept away the field and certainly became one of the preeminent people in modern music and other such things. So I'm not suggesting that Tito, whom you are replacing, is Bruno Volter or anything like that, but it's really a great attribute, and we're very grateful to Mauricio for just getting off a plane yesterday from a meeting in Panama, and he's going to discuss some very exciting work which he's been doing. He graduated from medical school in Chile, has been here at NIH only for 2 years now, or close to 2 years. He's a fellow in oncology and has done remarkable work. His bibliography and all is very impressive since he's been here. And we're very grateful to you for both substituting and on your own. And so people may ask you questions, hopefully, in the middle of your presentation. This isn't so formal, and we welcome you to do it. So some of the questions that go through my mind, and I put them up only to stimulate those of you, particularly students and fellows who don't think of this all the time, is some of the old problems. Is drug resistance intrinsic in a cancer cell? Is it due to clones that survive after therapy? And if so, really, how does that happen at a fundamental level of cellular and molecular biology? And on the other side of the bridge, how do you determine whether a cancer is responding, a solid tumor, whether it's responding to therapy? How do you do this in vivo? How good are the measurements that are made? And we're going to hear more about that. And I suppose the big question is, why are responses in solid cancers? Maybe there are some exceptions, but for generally, they're not curative. Why is it? If we step back and take a look at it in the big picture, which is what we're going to hear this afternoon, hopefully there will be some new thoughts and ideas, and from you as well. This is perhaps maybe the single biggest problem in oncology today. I had one other slide, which I'll just show briefly, and those of you who were here during John Coffin's elegant discussion of HIV a few weeks ago may recognize this slide. This was the point that the HIV virus decays in basically 4 phases after starting somebody on antiretroviral therapy. And you notice way over in my far left, phase 4, those cells which are as little as 50 or less per milliliter of blood, this is while a person's on multi-drug suppressive therapy, those cells persist. And there's an infinity sign there because nobody knows how long they persist, nor for that matter have they been clearly identified. But, whoops. Oh, well, I managed to shut it off. Oh, there. But if you stop therapy, the white bar, it's this clone of long-lived cells that contain an integrated retrovirus that go way up when therapy is interrupted. Very interesting. And you sort of wonder, and I just would throw this out perhaps for some discussion with our speakers, whether drug resistance clones, in the case of cancer, they require a new drug to try and get around it, I guess. But is there a similarity to these long-lived HIV-infected cells, that maybe there's something intrinsic in the cell? For the HIV, it's the integration site near responsive to a variety of promoters. But is it conceivable there's something like that in a cancer cell that actually renders it drug-resistant or long-lived? At any rate, you didn't come to hear me. So the first speaker is Michael. Thank you very much.
Thank you, Wynn. Let me just say by way of clarification that I have been at NIH, this coming since 1976, but I've been deputy for intramural research since 1993, which is long enough. It's over 20 years.
Did we, I thought we met in 76, is that right?
We may have, yeah, but I was not yet, no. So I'm always struck by the image that Wynn shows of this bridge, the Brooklyn Bridge, as it's being constructed, and part of the roadway is not yet complete. When we began to work on drug resistance in cancer, we discovered a gene which was responsible for pumping out anti-cancer drugs and had a very broad specificity or lack of specificity, and we said, aha, we have a major cause of drug resistance in cancer. And we thought the roadway was almost complete, and clinical studies began and so on. I would say now, 25 years later, we've removed some of the pieces of the roadway, and there are bigger gaps between the science and the translation. And I think both I and Mauricio will tell you what the problems are. But of course, with a better understanding comes some wisdom, and it certainly comes the kind of modesty that we really should use in approaching these difficult biological problems. So what I'd like to do is sort of give you some background information, give you a sense of where the field is, and then we'll answer questions, obviously, and then let Mauricio give you some more detail about some real clinical situations and how the clinical information we've gathered helps us focus on one or more of the different models that have been proposed to explain drug resistance. So if you fall asleep, and it is late in the day, I'll understand if that happens. What I'd like to point out is that this slide probably summarizes everything you need to know about drug resistance. First thing is that, as you know, oncologists have been very interested in developing drugs that are targeted to specific oncogenes. We've been sequencing cancer genomes now for many years. And in many cases, we know what the driving genes are that are responsible for the cancer. And the argument is if you target those specific gene products with specific drugs, and Big Pharma is happy to produce these drugs, that you should be able to cure cancer by blocking the essential growth-promoting pathway. This turns out actually to work reasonably well in chronic myelogenous leukemia, where there's a tyrosine kinase, the BCR-ABL receptor on the surface of the cell that drives the cancer. And there don't seem to be a lot of other ways that cancer can exist. And so if you inhibit that, you get very long-term responses. Although in some cases, resistance develops. And when it develops, it's due to alteration in the target, in the tyrosine kinase, or in pathways that are downstream from the target that bypass the target. So this is what I would call a fairly straightforward problem in drug resistance. And it works in understanding resistance of CML to this drug imatinib, but it turns out not to work very well for almost any of the other targeted drugs where you see resistance develop, and it's not usually this straightforward. So there are other ways for resistance to develop to multiple drugs, which is called multi-drug resistance. And some of them, I would say, are in the realm of cellular pharmacology or organismal pharmacology. That is, the drug either doesn't get into the cell or gets pumped out of the cell, or is metabolized in the cell, or is metabolized in the body, or is excreted in the body. All of these are pharmacological mechanisms that affect pharmacokinetics, pharmacodynamics of drugs that we use to treat cancer. And when we started our work in the 1980s, not very much was known about these mechanisms. And we were able to discover that energy-dependent drug efflux is an important mechanism for handling a lot of the drugs that are anti-cancer drugs. But uptake and metabolism and a variety of other cellular pharmacologic techniques are important as well. And finally, and this, I think, this kind of wisdom has come from actually observing patients in the clinic who are responding and then failing to respond to chemotherapy. Resistance can result from what I would call changes in the lifestyle of the cell. So a cell which has been driven to grow by virtue of one growth-promoting pathway, that's blocked by an anti-cancer drug, and so the cell simply chooses another growth-promoting pathway. Or there are changes in pathways that are normally associated with differentiation, and one which has been very much studied in this area is called EMT, or epithelial to mesenchymal transition, where a cell that looked epithelial is now growing as a sort of more mesenchymal stellate cell, And those cells have totally different patterns of sensitivity and resistance to drugs, and that drives their resistance patterns. Or, in many cases, resistance can develop because we may be dealing with a tumor that looks to be homogeneous to begin with, let's say breast cancer, but there are really 4 subtypes that are very different molecularly, although not necessarily histologically. And those subtypes each have different patterns of sensitivity or resistance to drugs. Or a tumor cell can develop along pathways which are starting from the same cell but are somewhat different depending on the initial initiating factor. So it's the biology really that determines, in this third case, that resistance developed. And it's this kind of problem that has led many people to kind of throw up their hands. Mauricio and I were at a symposium on physical science and oncology just a few days ago at NIH. And we were on a panel in which a number of members of that panel said the problem is insoluble because it's intrinsic to the nature of the biology of tumor cells. We don't believe that. You're not going to hear that negative a message today, but I think hopefully we'll convey to you some of what the complexity is of this problem. So there are certain factors that lead to the development of resistance, and one which I think has only in the last few years been fully appreciated. Because, I mean, you know, remember when we learned about cancer, we were taught that there was a single cell that underwent malignant transformation, and that cell grew as a clonal population to develop a fairly homogeneous population of cancer cells. And if you believe that, then you believe that you should be able, with one drug or combinations of drugs, to wipe out all those cells because they're so similar to each other. Well, the reality is that if you look at cancers in detail, in molecular detail, including sequencing of the DNA of individual cells as a cancer, each cell is quite significantly different. And they're different for a variety of reasons. Some of it has to do with the fact that there are just mutations arising in these cancers. Some of it has to do with the fact that the expression of genes in those cancers changes, there are epigenetic changes. And some of it may do with the fact that the drugs we use to treat cancer actually induce mechanisms of resistance because life would not survive if we didn't have really good ways of dealing with toxic products in the environment. So all of these add to the difficulty when you're starting with a fairly large tumor population in finding a drug or a combination of drugs that will kill all of the cells. So my talk will have 4 parts, and I'll go through this pretty quickly. The first part has to do with these transporters that we've been working on now for 25 or 30 years, what role they play in drug resistance. Secondly, a kind of technical note about one of the reasons that we've had these moments of epiphany thinking we've understood drug resistance and then don't, and that has to do with the model systems we're using, which are not always relevant systems for studying cancer. Then I'll talk a little bit about some work we've done, sort of preliminary work we've done, looking at expression of drug resistance genes in clinical cancers to show you that even with a simplifying assumption that the genes we're interested in are important genes, that the complexity of actual tumors is something quite amazing. And then a little bit talk about models to introduce Mauricio's talk that might be important for clinical cancer. So this is just a summary of what I already said to you in terms of cellular mechanisms that could lead to resistance. Just to give you some idea of the scope of this problem, we know in the human there are 386 transporters that are called solute carriers, or SLC. If you look at gene descriptions, you always see SLC, XYZ. SLC means solute carrier. There are 386 of them in the human genome, and plausibly about 10% of those, or on the order of 40 of them, are involved in the uptake of drugs into cancer cells and into normal cells as well. Some of the toxicity of anti-cancer drugs probably relates to the expression of these genes in specific organs or tissues that are sensitive to the drugs. And it appears that in order for a cancer cell to be sensitive to a specific drug, the drug needs to get into the cancer. We used to think it was simply a matter of a hydrophobic drug dissolving across the membrane, but more and more evidence exists that there are specific transporters. And when those transporters are expressed at higher levels, it's easier for drugs to get in. So they can determine the sensitivity or resistance of tumor cells or normal tissues to drugs. There are also these efflux pumps, which were discovered, as I mentioned, around 25 years ago. And these include a number of ABC, meaning ATP binding cassette transporters, and I'll be talking quite extensively about those in a moment. Several of these are involved in export of drugs from cells, and in the same way that uptake transporters can keep drugs out or let them in, efflux pumps also reduce the accumulation of drugs in the cells. And then if a drug does get in, and frequently drugs can, and because the pharmaceutical companies are always looking for agents that can get into cells, there are a whole variety of changes that I'll go through in more detail, involving regulation of cell growth and apoptosis. Apoptosis is programmed cell death, metabolism of drugs, altered targets, repair of damage, and this is particularly important. A lot of anti-cancer drugs damage DNA, and there's a whole set of really important DNA damage repair systems that play a role in drug resistance. And compartmentalization, which sometimes for fun I call sequestration because it's such a terrifying word. At least in the federal government it is. So what I'm going to do now for a while is focus on these efflux pumps, not to give short shrift to any of these other mechanisms, which I'll get back to in a moment when I show that this is not the single most important explanation for drug resistance. So the ATP binding cassette family is a big family. There are 48 different varieties in the human. If you look at all published results, there are about 2,000 members that have been identified, many of which have unique characteristics. The major one is that they use ATP to pump drugs and other compounds out of cells. These include ions, sugars, glycans, et cetera, this long list of things, and natural product anti-cancer drugs. Structurally, they consist of combinations of these ATP binding cassettes and segments with 6 transmembrane domains. And these are shown here. This is the family of human ATP binding cassette proteins. And this is a figure made by Mike Dean, who's been studying the ontogeny of these transporters. And you can see some of the transporters consist of these 6 transmembrane domains and an ATP binding cassette. The canonical form of transporter has 2 of these 6 transmembrane domains and 2 ATP binding cassettes. But you see combinations of various types, and I'll show you some more examples in a moment. And there are 7 different varieties, families of these transporters, and drug resistance transporters exist in several of the different families. So there's nothing unique about one of the families. The families are determined mostly by homology within these transmembrane regions, since the ATP binding cassettes are pretty similar throughout all the different members, and that's what makes this a family. There's some features that are kind of interesting. Because there's a fair amount of homology, these are hard to individually detect using standard techniques like microarray, and I'll say more about that in a moment. So I'd like to focus on 3 of these transporters, and I chose these because these are the broadest spectrum multi-drug transporters. If you select for cancer cells and tissue culture that are resistant to a whole variety of drugs, these are the transporters that tend to come up. ABCB1, which has been called for many years P-glycoprotein. This term was coined by Victor Ling, where P stood for permeability, and now P stands for pump, because it's not a drug that regulates what gets in. It's not a transporter that regulates what gets in. It's pumping things out of the cell. And this has these transmembrane domains and 2 ATP binding cassettes. The ABCC family, of which MRP1, multi-drug resistance protein 1, is an example. Some of those have this amino terminal extension of 5 transmembrane units. You can cut this off, and it's still an active transporter. We're not sure exactly what this is doing. It may have something to do with cell localization. And then there's the ABCG2, which looks like a half molecule, except that the ATP binding cassette is on the amino terminal side. And this undoubtedly works as a homodimer to form 2 regions with a total of 12 transmembrane domains and 2 ATP binding cassettes. So we think the basic mechanism is similar for all 3 of them. Their specificity varies a little bit, as shown in this Venn diagram. So ABCB1 recognizes all of these substrates and many, many more. C1 recognizes these, and G2 recognizes these. And they all recognize these drugs in common all of which are anti-cancer drugs. The anthracyclines, doxorubicin, daunorubicin, mitoxantrone, etoposide, and nilotinib. Those are shared in the human. If you look in other organisms, for example, the mouse, you see somewhat different specificity. And in fact, the mouse has 2 ABCB1 genes, so it has 4 gene products that are involved in multi-drug resistance. All of which is a complexity that makes it difficult for companies that are using engineered mice and so on to be able to study this problem a little bit at a loss. So if you look at the 1,280 amino acids that make up this transporter, in a planar structure, it looks something like this. The yellow represents specific point mutations in P-glycoprotein that change its substrate specificity. So even though it recognizes literally hundreds of different drugs, you can make a mutation in any of these sites and you get changes in the drugs that are recognized, and we're just beginning to try to understand why that is. There's a region called the linker region, which is phosphorylated, but the phosphorylation doesn't seem to regulate function. You can eliminate the sites or dephosphorylate it, and it still works as a transporter. And these green bars represent, if you do affinity labeling studies with drugs, you end up labeling the transmembrane regions, which led us to believe that both regions were involved in drug binding. If you knock out either ATP site, you knock out transport function. And so we thought these 2 parts probably came together, and we came up in the 1980s with this model based on the sequence and some of the biochemistry we had done. And the most amazing thing about this model is that if you take, and I don't have with me a high-resolution EM, cryo-EM picture, it looks exactly like this. P-glycoprotein actually looks like this at a resolution of about 10 to 20 angstroms. And so what we think is happening is that because of the hydrophobic nature of the drugs, that they're actually being recognized within the plasma membrane. The ATP is hydrolyzed, and I'll show you a little more detail in a moment, and these drugs are being pumped out. The important thing of understanding that the drugs are recognized in the bilayer is that explains the lack of specificity, that there are hundreds or thousands of drugs. And the essential feature has to do with the hydrophobicity of the drug, some other features as well, but there's no clear pharmacophore. It's a physical property that determines that it's a substrate for this pump. These are higher resolution pictures that are based on some crystallographic studies that have been done. The mouse MDR-1A gene, which is a homologue of the human ABCB1 gene, has been crystallized by Steve Aller and Jeff Chang to relatively low resolution, but they were able to crystallize the protein with a drug bound. And this is the bilayer here, and you can see the drug is indeed bound in the lipid bilayer. And it's bound in a region which is the same region that I defined before as being the part of the molecule which is photoaffinity labeled. So the notion here is that the drugs get into a hydrophobic pit. They have some specific interactions with amino acids depending on the nature of the drug. When ATP is bound, the 2 halves of the molecule come together. This is called the open conformation. This is the closed conformation. And what that does is extrude the drug from the transporter. And when ATP is hydrolyzed, it resets the pump so it can pump again. The precise details are being studied in a number of labs, in our lab particularly by Suresh Ambudkar, who could give you a little more detail about how this actually works. So when we first discovered that P-glycoprotein was a major efflux pump in cancer cells that were drug resistant, we said, well, where did this come from? This must be expressed normally in tissues. And in fact, it is, not uniformly in the body, but in certain tissues that have barrier functions. So it's present in the small and large intestine, pumping into the lumen of those organisms. It's present in the liver, in hepatocytes that are lining the biliary ductules. So it pumps into the bile, pumps into the intestine. In the kidney, it's in proximal tubule cells, pumps into the proximal tubules and into the urine. In cells, and I have here circulating CD4 cells that express P-glycoprotein. And just to get back to what Wynn told you about earlier about the resistance of some of these the patients to HIV ART therapy, antiretroviral therapy, some of those drugs that are used to treat HIV, particularly HIV protease inhibitors, are really good substrates for P-glycoprotein. And there's a fair amount of evidence that there are subsets of cells that circulate that express P-glycoprotein or ABCB1. And that some of the resistance of the virus may be due to cellular resistance to these compounds. And then there are cells that are in the interstitial space, in particular, in this case, tumor cells that can express PGP. The other place, which I want to talk about in a moment, where PGP and these other multi-drug transporters is expressed, is in the brain, in capillary endothelial cells that use energy to keep hydrophobic toxic compounds out of the brain. And so what basically this says is that for P-glycoprotein and for the other transporters as well, maybe I have a, no I don't, that basically the PGP normal function is a barrier function. It it puts things out of the body and keeps things from getting absorbed into the body. So just a couple of words about the blood-brain barrier, because this turns out to be rather important in treating brain tumors, for example, getting drugs into the brain. And a lot of people who are studying neurobiology and trying to cure degenerative diseases of the central nervous system are trying to get things into the brain, and a lot of drugs don't get into the brain. And why is that? So if you look at this graph, which shows the octanol solubility of drugs, that is their hydrophobicity, and along this line they get more and more hydrophobic, they dissolve better in octanol. And compared to uptake into the brain, you can see that a lot of compounds behave fairly normally. That is, the more hydrophobic they are, the more they get into the brain. But some compounds fall off this curve, and those include glucose, which gets into the brain quite readily, despite the fact that it's not very hydrophobic. And the reason for that is, clearly, there are glucose transporters that pump glucose into the brain. And then there are all these drugs, bleomycin, adriamycin, epipodaflotoxin, cyclosporin, vincristine, that fall off the curve in this direction, that are anti-cancer drugs and hydrophobic natural products that, despite the fact that they're quite hydrophobic, don't get into the brain. And the reason they don't get into the brain is because of this blood-brain barrier, which is made up of, first of all, the capillary endothelial cells form tight junctions, so things can't leak into the brain. And then there are these pump systems. Whenever anything tries to dissolve into the brain, it gets pumped back. So the same system that protects cancer cells from hydrophobic toxic anti-cancer drugs seems to be involved in this important blood-brain barrier. And this is just another picture showing the hydrophobic vacuum cleaner. Stuff tries to get into the cells, it's recognized and pumped out. So Matt Hall in the lab has been particularly interested in understanding how to image the blood-brain barrier with the idea of being able to circumvent its activity, particularly if you want to get drugs into the brain in neurological diseases and also, for example, in cancer. And so we set out with Bob Innes and Victor Pike, who are colleagues in NIMH who work on PET imaging, positron emission tomography. Has anyone talked about PET yet here? Well, I'm not going to talk about that. So. Yeah. Anyway, it's a very sensitive way of detecting molecules in different parts of the body. And so we made a derivative of a compound called loperamide. Loperamide is what you buy over the counter as Imodium. It's an anti-diarrheal drug. And the reason it's anti-diarrheal is it's a potent opiate. And the reason it's sold over the counter is it doesn't get into the brain, so it's not intoxicating. So you don't get addicted to it because it can't get into the brain. And the reason it can't get into the brain is it's a substrate for ABCB1 or P-glycoprotein. Quite a good substrate. And you can see here, these are cell lines in which PGP is expressed or not expressed. ABCG2 and C1, the other multi-drug transporters, are expressed. And only in the presence of ABCB1 do you see this big reduction in accumulation of this compound, which we PET labeled. If you look at knockout mice that are lacking ABCB1, you see a big increase in uptake in the brain of this compound, D-loperamide, whereas in the G2 and C1 knockouts, you don't see uptake. So this is another bit of data that suggests in the mouse that uptake is dependent on B1. And so you can use a C11 derivative of dimethoxyloperamide as an imaging agent injected into mice. If you do that, you see that this is the mouse brain. And just to show you, this is a CT scan. The mouse has a small brain, but it has a brain. And this is the brain. And there's nothing in the brain until you add an inhibitor of P-glycoprotein, which in this case is a compound called tariquidar. And then you get nice uptake of material into the brain. So that shows that the system is working. You can do the same experiment in a monkey. You can see dramatic uptake in the brain. In the human, you do see an increase in uptake. It's not as dramatic as it is here because the drug that we use, tariquidar, we can't give in high enough doses, not because the drug is toxic, but the agent used to dissolve it is fairly toxic. And so we're still working on finding a better way to deliver it. But you can see clearly substantial increase in uptake in the brain. And this actually points out how difficult it is in the human to totally block P-glycoprotein expression. You need pretty high levels of potent inhibitors, which is one of the reasons why some of the clinical trials to reverse drug resistance probably have not been so successful, because they haven't done a good job of reversing PGP expression. So I just thought I'd end this part on P-glycoprotein by summarizing the data that we currently have that does support a role for P-glycoprotein or ABCB1 in cancer. The first thing is that it's expressed in human cancers at levels that are quite sufficient to confer drug resistance. So at least 50% of human cancers express reasonably high levels of PGP. There's correlative data showing that after you treat certain tumors with drugs that are substrates for P-glycoprotein, the recurrent tumors, the relapsed tumors, express higher levels of P-glycoprotein. This is true for leukemias, myelomas, lymphomas, and breast and ovarian cancer. You can, in some cases, for example, acute myelogenous leukemia, show an increased response to anti-cancer therapy if you inhibit P-glycoprotein. But for many of these tumors, that's not the case. And even though PGP goes up, inhibiting it doesn't solve the resistance problem. There are a bunch of tumors which express PGP at the time of diagnosis, including those derived from cell types that normally express PGP. Remember I mentioned in the small and large bowel, PGP is expressed. Colon cancers express PGP. Kidney cancers express PGP. Pancreatic and liver cancers. So these are tissues that normally express PGP. When they become malignant, those cells continue to express PGP, frequently at quite high levels. And those are intrinsically resistant tumors. And then you can make animal models with human cancer xenografts or models which are endogenous to the mouse, in which, for example, you can use P53 mutations and BRCA1 mutations to get breast cancers in mice. And when you do that and select for drug resistance, you get PGP expression, and you can inhibit PGP expression, and you get response to the tumors, and then they grow back and they're resistant, but not because of PGP. So the conclusion of all of this, and the reason why I said the roadway is getting more and more, roadway was nice and clear until we started doing studies. It's the problem with getting facts. You really should avoid it all that you can. Is that P-glycoprotein is sufficient to confer drug resistance. Every model suggests that. I didn't show you we have a transgenic mouse in which we put in PGP. Those cells that express it are drug resistant. But it may not be necessary. In other words, there are many other causes of resistance. Or when it's expressed, it may not be the only resistance mechanism expressed. So that's the problem. That's the clinical problem. So we went back to the drawing board and we said, okay, it's not going to be PGP, and since we're interested in studying drug resistance, we should try to figure out what it is. And we went to the literature and we found about 380 drug resistance genes that people had described over the last 20 or 30 years. We talked to our colleagues and we put together a set of these genes using a system called the TACMAN low density array to measure very accurately and precisely the RNA in both tissue culture cells and tumor samples for each of these 380 genes. And the genes involve in uptake of drugs, sequestration of drugs, metabolism and efflux of drugs, genes involved in pathway of ceramide synthesis, which seem to be involved in drug resistance, pathways involved in uptake of drugs into the nucleus, DNA repair, and all of these genes involved in regulating cell growth and so on. And we had 380 genes, and we could easily distinguish them. We could measure them with high specificity over a long linear range and at very low levels of expression. And so the first thing we did was we said, well, let's take a look at our cell lines that we've been using to study drug resistance and see whether those are good models for it actually exists in tumors. So we got tumor samples. We started with ovarian cancer for a reason I'll tell you in a moment, because it seemed to be a good model for both intrinsic and acquired resistance. It's a disease which responds to chemotherapy, but all too often the patients get resistant and unfortunately die of their tumors. So we got a lot of ovarian cancers, and those are shown here. This is a heat plot. These are the 380 different genes, the level of expression. Red means high level of expression, green means low level. And these are about 100 different ovarian cancers. And what you look for is the pattern of gene expression to see whether the ovarian cancers actually have patterns of gene expression similar or different from the cell lines we're using as models. And you can see they're pretty similar to each other. These are ascites tumors, which is a slightly different histological kind of tumor. And these are the cell lines that we've been using, 40 different ovarian cancer cell lines. And their pattern of expression of these 380 genes is totally different. We've been depending on these to understand drug resistance, but they don't seem to be representative of the tumor cells that are coming out of the patients. So we said, is this a kind of general problem or one unique to ovarian cancer? So we looked at melanoma. These are the tumors. These are the cell lines. We looked at breast cancer. These are the tumors. These are the cell lines. We looked at gliomas, tumors in the brain. These are the tumors. These are the cell lines. Colon cancer, interestingly different patterns, but these are the cell lines. These are the tumors. So essentially every tumor we looked at, with maybe the exception of hepatoma, and Wynn and I have had this discussion before. Hepatoma is a tumor derived from the liver, and I think the liver cell already expresses a lot of these interesting drug resistance genes, and so when it gets established in cell culture, it doesn't change its mode of behavior. It still continues to express the genes, whereas these other cell types probably need to adapt to tissue culture. So this basically says that our current models are not very good. We need better models. Why do we need better models? Okay, so when we establish cells in tissue culture, and I have to say that a lot of the cell lines that we use have been in tissue culture almost forever, literally longer than I've been in this field, 30, 40, 50 years. And so they established themselves in tissue culture. Many people think human cells grown for 30 years in culture should be a different species. Everything about them is really different. Gene expression is very different. And they're particularly different with respect for these genes that we're studying that relate to environmental response to environmental adversity. So we think that what survives is probably either a small subset of the original tumor, or more likely something which is by mutation and adaptation changed its way of living by being in tissue culture. The other thing about culture, the way we culture most cells in monolayer culture, is that the culture conditions are very different. We use room air when the oxygen tension in tissues is much lower. We grow them in monolayer generally versus 3D culture. In tumors, there are other cell types, and there's more and more evidence that a lot of the behavior of tumors relates to the other cells, so-called normal cells, which are probably not so normal, that surround the tumor. And of course, growth factors. And finally, we're not happy unless we grow cells very quickly in tissue culture. We want them to double every 12 hours, every 16 hours, maybe every 20 hours. Normal tumors don't grow that quickly. Most of the time, they're not growing, because if they did, can you imagine how very quickly those tumors would totally overwhelm us? So we set up conditions that are quite abnormal in terms of forcing the cells to grow. And of course, it's not surprising that they express genes that are very different from those that are expressed in normal tumors. So all of these things work against us. And so we decided we would try to start developing a better system for growing tumors, more like organ culture, ex vivo from the body. Take pieces of tumor and try to grow them under conditions that were as close as possible, and then study drug resistance under those conditions. And so I was very fortunate to have a postback in the lab, Ashley Jaeger, who was a bioengineer as an undergraduate, is now in medical school. And we worked with Pohida Pohida and his colleagues at CIT who were bioengineers. And the idea was to develop artificial capillaries so that we could create oxygen gradients that were similar to what you see in tissue. So you imagine a capillary running through, oxygen and nutrients are running through the capillary. Those are diffusing away, and as the further you get, the less oxygen there is. And the starting concentration of oxygen is actually quite low. So the idea was to use materials that would allow oxygen to diffuse, and we initially came up with silicon hydrogel. For those of you who don't know what it is, if you wear contact lenses are made of silicon hydrogel. They're easy to mold, and they're very permeable to oxygen, which is why they're used to make. And so we made these things that are capillary size. This is a photolithography technique that Ashley developed. And the idea is that oxygen in this chamber diffuses up from the bottom. It goes through these artificial capillaries and diffuses away and forms gradients that are similar to what you'd see in normal capillaries. We've confirmed that. This shows how cells grow in this system. They grow around these artificial capillaries. If you eliminate the gradient by having the same oxygen concentration in the top and the bottom, they grow as spheroids. If you don't have the pillars, they grow as spheroids. Spheroids are these little round tumor nodules that are the typical way that tumors grow in 3 dimensions. So their growth is quite different. And if you look at an individual tumor growing in the capillary and use a compound called pimenidazole, which is a histochemical stain that detects lower oxygen concentrations. So the staining is for hypoxic tissues. You can see a gradient of oxygen away from the capillary, which mimics the actual oxygen gradient in these systems and is very close to what you see in actual tissues in artificial capillaries. So we think we have a reasonably good system. We're just starting to look at real tumors in this system. And we hope to be able to measure gene expression as a function of distance from the capillary. You can see immediately another reason why tumors are heterogeneous, because oxygen, which is an amazingly important determinant of gene expression and gene regulation, concentrations are totally different throughout the parts of the tumor. So that's one of the drivers probably for heterogeneity. Okay, so very quickly, we've been looking at 3 different tumor types representing intrinsic or acquired resistance. So let me just say intrinsic resistance is a tumor that just doesn't respond to chemotherapy to begin with. Acquired resistance is the tumor seems to respond and then relapses with resistance. And we never knew, and I'm not sure I know now, whether these are different mechanisms or 2 sides of the same coin. I think Mauricio is going to make an argument that acquired resistance is actually a kind of intrinsic resistance, that the seeds of acquired resistance reside within the initial tumor. Hepatoma doesn't respond well to chemotherapy, and acute myelogenous leukemia, which mostly does respond, but in many patients relapse and become resistant. And the advantage of studying this tumor is because it's a liquid tumor, we could get samples from patients both before and after therapy in the same patient, which is very difficult for the other tumors. So what we did was we were very fortunate to have an outstanding biostatistician. We accurately measured expression levels for 380 genes, and we said, are there any genes whose expression in ovarian cancer predicts response to therapy? We knew the response in the various tumors. And came up with 11 genes, all of which were important statistical contributors. Together, they were the best predictors of whether the tumor would respond or not to therapy. Just to remind you, therapy in ovarian cancer, most patients present with pretty advanced cancer, stage 3 or 4 disease in the abdomen. The therapy consists usually of a Taxol-like compound, a taxane, Taxotere, or Taxol, and a compound which is platinum-related, cisplatin, carboplatin, and so on. And therapy is pretty much the same around the world. And so each of these genes represents different mechanisms of resistance. Glutathione peroxidase is a detoxifying gene. There are apoptosis suppressing genes. There are growth factor promoting genes. There are kinases involved in growth factor pathways. There are genes involved in antigen presentation, genes involved in metastasis and invasiveness. All of these together turned out to be a good way of predicting. So we can predict. If a patient comes, we can say the likelihood that they'll respond or not based on these gene expression patterns. Now, the fact that we could find a subset of genes, not a single gene, suggested that either ovarian cancer is multifactorial, that is, all of these resistance genes are contributing, or that all of them simply represent a pattern of resistance, but there's one predominant gene which could be different in different tumors, and we would not see that with the statistical approach that we took. A third possibility is that cancers with different gene expression patterns arise from different origins. Those of you who know anything about ovarian cancer know there's a big argument now in the literature about the origin of ovarian cancer, and it's possible that cancers from one origin are more responsive than cancers from another origin, and the gene expression patterns differences we're seeing relate to the origin of the tumor or the way it became a cancer. So just quickly, in hepatoma, there's a 45 gene signature. I showed you an 11 gene signature for ovarian cancer. It's more in hepatoma. That distinguishes better prognosis from worse prognosis tumor. We've independently confirmed this. And again, this suggests that there are either two different cells of origin or different ways in which hepatoma can develop. And in AML, we know that P-glycoprotein expression has been shown to correlate with poor response. But when we look at different paired samples before and after, we see increased expression of PGP, but increased expression of G2 or increased expression of C1 or any of the other ABC transporters or any of the other drug resistance genes. Every tumor we look at that's become resistant has a different pattern of gene expression. So we suggest that the basis of resistance may be somewhat different in each cancer. This is the argument for personalized medicine, but this is the depressing part of the study. If every patient has a different reason for resistance, it's going to be pretty hard up front to figure out what to do about that. And we'll have to wait and see what develops, and then we'll have to work with that information. So just very quickly, a couple of models to account for what we're seeing here. In acquired resistance, what we're saying is we're using two different drugs to treat ovarian cancer, a blue drug and a green drug. Most of the cells seem to be sensitive to the blue drug and the green drug, but there are some resistant cells in this heterogeneous population to begin with. If you use drug A, the resistant cells are blue, and also mixed in here are some multi-drug resistant cells that are just resistant to everything. Same argument with drug green. And if you add them together, which is what we do in ovarian cancer, you can kill most of the cells, but the ones that survive may be the multi-drug resistant ones. And similarly, in terms of intrinsic resistance, you can say that up front you have a heterogeneous population of cells. Some of the heterogeneous populations have more sensitive cells than other ones. So, for example, in this population, there are a lot of resistant cells, but in this population there aren't too many resistant ones. You get a good response, but unfortunately in a lot of the patients the tumors grow back. So just a final thought, because I think this may stimulate some discussion if you're interested. So we've been using successfully natural product anti-cancer drugs to treat cancer. In fact, the most successful drugs we have are natural product drugs, even though a lot of advances have been made in more targeted drugs. So the idea here is that these drugs have evolved. They're made by plants and microorganisms, and they've evolved over the years to kill cells. This is part of the warfare between plants and animals. So they know how to kill cells. We're not exactly sure how they work, and one of the things we talk about is not so much what the basis of resistance is, but what the basis of sensitivity is. But they can kill cells. They target multiple pathways that have also evolved over time to preserve life in the face of extreme environmental conditions. So we are alive because we have a lot of mechanisms for resisting these toxic things in the environment. In our cleverness to create targeted drugs, we're targeting single pathways, and that these generally don't work to cure cancer because there are all these compensatory mechanisms that allow some subset of cells to survive even in the face of this kind of onslaught. So that's just a philosophical thought. Maybe Mauricio would want to comment on that as well. I have a lot of people to thank, and I'll just say that the clinical work was done mostly with the help of Jean-Pierre Gier, who was a staff scientist in the lab. We have a lot of clinical collaborators. Sudhir Varma was responsible for the biostatistics. And I've had the good fortune for many years to work with Suresh Ambudkar. T. Shah is a crystallographer, and other people in the lab have contributed in too many ways to count. So I'm happy to end there, and we can either take questions now or wait till after Mauricio's stuff.
Thank you very much, Michael.
Yes? My mic is on. Oh.
Okay.
My name is also Mike. That's why I was confused.
Wait a minute.
You mentioned that the MDR, although you pointed out so many other mechanisms, but MDR still is a focus of a lot of our attention. And you mentioned that it is clearly sufficient to generate resistance. So what do we know about regulation of expression of MDR, and how can we address that issue?
Right. So we know quite a lot. Let me just add that the main way in which MDR has influenced anti-cancer therapy is because pharmaceutical companies screen all their new compounds against MDR cell lines. And they always hope to find compounds that will get into cells that won't be able to be pumped out by MDR. So a lot of the drugs entering the market are already screened to not be MDR substrates. So that's a big contribution in an important way. So that eliminates P-glycoprotein as an important mechanism. In terms of what we know about regulation, so obviously there are two kinds of regulation. The regulation that's developmental, so these tissues that I mentioned, the epithelial cells of the GI tract, proximal tubule cells in the kidney, the hepatocytes, capillaries in the endothelial cells, and it's in the placenta, it's some other places. All are developmentally regulating expression of PGP. And there's a little bit of information about the transcription factors and the upstream sequences that are essential for regulation under those conditions. In addition, there's pretty good evidence, both certainly in tissue culture and also in actual tumors, that PGP is acutely induced by toxic compounds. And not only substrate compounds, so compounds that damage DNA result in PGP expression. So it seems to be under control of these orphan receptors that are involved in turning on genes that protect against toxins. And again, a little bit is known about the binding sites in the upstream promoter region and so on. It's kind of a very interesting area because if it turns out that resistance is due to simultaneous expression of many different resistance mechanisms, and those are under relatively universal control, there's a single transcription factor or there's a couple of factors, those then become really interesting targets for regulating response. And not enough is known to know whether that's true or whether the regulation is going to be as complicated as the genes that promote resistance. It's a good question. John.
Why do normal, good hydrophobic substrates not interfere with what you're trying to accomplish in terms of poor specificity? So cholesterol, or even maybe good cholesterol, big lot of cholesterol is good for you. Tell it to the cardiologist.
Okay, so it's a really interesting question. So the specificity is broad, but obviously there aren't too many. I always get asked, it can't be that the natural substrates for PGP are all exogenous compounds. There must be some endogenous compounds that are substrates. So there's a whole field that believes that cholesterol is in fact an important component of the structure of P-glycoprotein. You don't see it in the crystal structure, but the resolution isn't quite high enough to see it. That PGP is recognizing and binding cholesterol as part of its, because it does recognize steroids and pump steroids, for example. So statins are wonderful substrates for P-glycoprotein. So the answer is probably in evolution it was important that it not pump normal compounds that are present in the lipid bilayer, but recognize things that were abnormal. I mean, I think it's a kind of surveillance system for making sure that bad stuff doesn't get into the membranes. once we settled on having membranes, we wanted to be sure that we could clean those. That's why I call it a hydrophobic vacuum cleaner, which is a little bit controversial. But the other point is that every one of the compounds that binds to or is transported by PGP is a potential inhibitor of PGP or modulator of PGP. And so there are a million different inhibitors. There's no lack of inhibitors. And anything which is non-toxic, which is a substrate, becomes a non-toxic inhibitor. So that's why we know that just inhibiting it isn't sufficient, probably, to reverse drug resistance in cancer.
I mean, it's interesting that 9 of the 10 top candidates for causing acute liver injury in man are antibiotic derivatives of microorganisms, which don't affect the organisms.
And some of them are cytochrome P450 substrates, some of them are PGP substrates, some of them are both. Actually, cytochrome P450, which metabolizes drugs, the 3A4 derivative, has a drug specificity which is almost identical to PGP. The overlap is enormous. So here's a transporter and a metabolizing enzyme, but there's no sequence homology at all and probably not much structural homology. So nature has solved this problem of recognizing these compounds at least 2 different ways.
Okay, but save your questions. We'll have more time at the end. And Mauricio, thank you very much, Michael.
Okay, the one that's wiped off.
Well, thank you for the opportunity to replace Dr. Fojo in this afternoon. And I will talk about the novel models for cancer that might lead to better treatments. And now in the clinical setting, I'm talking to treating patients. We can remember in the 60s and 70s was the exciting times for oncology and chemotherapy for lymphoma, mainly lymphoma patients, because Vincent DeVita here in Building 10 tried a combination of that cause responses in many lymphoma patients. And in other places, as in other institutions, start to be wondering what would be the best way to reach to the best combination of the best drug. And really, the problem starts to be comparing therapies. That's emerged as important goal or objective between institutions. And I want to tell this story that Dr. Fojo told me that I didn't know about it. Is all the RESIST and WHO criteria for clinical trials are coming from this doctor, Dr. Martel, in 1976. And he did an experience, really, an experiment, an experience in an ASH meeting, American Society of Hematology meeting, with 16 oncologists and 12 spheres and 1920 measurements. What he did is that were 12 spheres from 1.8 to 14.5 centimeters in diameter. And they asked to each oncologist in that meeting to measure these spheres. The thing is that 2 pairs of these spheres were equal in size, the number 5 and 6 and 7 and 8. But the oncologists that were not aware of that was the fact. And what happened at the 25% of the time when they have to realize that the difference was more than 25%, they did a mistake. And it was when 2 different oncologists measured this and it has to be more than 50%, just 6.5. That is logical because the difference between 2 spheres, 25% and 50% is larger. And then the same oncologist has to measure and did the mistake for this 50% only in 7% of the cases and in 25% in 19% of the cases. That is really the base of the WHO criteria for evaluation of response for solid tumors nowadays, with some difference that we'll talk about. And we can say that from these beginnings, from cutoffs and choosing for really practical reasons, but not really for efficacy or science, we evolved for the assessment of efficacy of drugs in oncology. And the WHO then was evolved to the RECIST criteria that we use now in clinical trials here and in many places, with some subtle difference in how we measure tumors, bidimensional measurements, and now unidimensional measurements of the tumor, or some difference in the criteria for, say, that a patient is progressing of his tumor, 50% versus 30%, but really are based on the same Martel experience. And here you can see the subtle difference between the final volume when you assess and you say that a patient has progressed. In the old criteria, you have to be a 140% increase or 40% increase in the volume of the sphere or in a tumor to say that a patient has progressed. And now with the new RECIST criteria is 173, that means 73% more. I mean, a more relaxed way to define progression. I mean, before you assign progression in a patient before than now, but really it's almost the same. It's just some evolution in the criteria. And that means it has been 37 years since Martel study, and the question is, should we be thinking about different ways to assess efficacy of drugs and targeted agents? That's the main thing. And I will start with the example of kidney cancer. That is the model that we choose, one of the models, to try to understand a better way to assess efficacy of drugs. And this is a simple diagram to explain how the new drugs for kidney cancer in the last 10 years has been envisioned according to all the mutations in the von Hippel-Lindau gene, somatic or germline, that cause the accumulation of this transcription factor in the cytoplasm, HIF, that cause increase in the transcriptions of VEGF, GLUT1, and many genes related to proliferation and angiogenesis. And one of the drugs that was first used for like a targeted agent was sorafenib, that is used and approved still by the FDA for kidney cancer. And it was envisioned as a drug that can cause a chronic, make kidney cancer a chronic disease. Disease stabilizing agent. But really, if you see the waterfall plots, this is each of these lines is one patient, and each line represents the best response of each patient. And you can see really 74% of the patients has some decrease in the size of his tumor. That means really decrease the size of the tumor. The thing is, the threshold for causing a partial response was not reached. And for that reason, it's called a stabilizing agent or cytostatic agent. But really, if you see the waterfall, you see there's killing of the cells in this drug. For that reason, in the group of Dr. Fojo many years ago, I started to think, how will be a better way to assess this difference between drugs, targeted agents, called targeted agents, and traditional agents. And this is the equation that Dr. Wilfred Stein is a physics and mathematician that works with Dr. Fojo many years, try to model the phenomena of progression and regression of tumors. And here you can see this is a plot of the y-axis is the tumor measurements that you can see in a daily basis in practice of clinical oncology, and this is time. And for example, if you have a patient that responds to a therapy, you have a regression in the tumor, the size and your measurements, and then resistance appears and you have a growth of the tumor and progression. And the patient, if is in a clinical trial, is put off the clinical trial. But we envision this in the way that this curve, this sigma plus curve, is composed for two components. A component that is the tumor that is sensitive to the drug that is under treatment, and a fraction of the tumor that is resistant from the beginning. And this is the concept that Dr. Gottesman was saying about intrinsic resistance. And between these two fractions, you can construct this curve, and this curve fits this equation, this mathematical equation that really is saying that the size of the tumor at any point in time if you follow in the patient under treatment, is an exponential function of the decay, how the tumor, the sensitive tumor decreases with treatment, and G, how the tumor is growing. And these two are constant, and the variables are really the time under treatment. The -1 is just a value for start the equation for the number 1, that is the start of the treatment measurements. And this is the first analysis that they did, I think like 7 or 8 years ago. This was a phase 2 trial done at NCI by James Young and Dr. Rosenberg in the surgery branch with bevacizumab, this is a monoclonal antibody, against placebo for metastatic renal cancer patients. And just for you that you don't know, bevacizumab is a monoclonal antibody that targets VEGF, that is a growth factor in the blood, that targets the receptors in the cell membrane and really impedes that this growth factor activates these receptors that are involved in angiogenesis. Later, we'll see that small molecules like sorafenib, sunitinib, pazopanib, or axitinib are also target the same receptors, but in a different way. And this is the Kaplan-Meier curve of that paper, the New England Journal paper of Dr. Zhang, that shows that the progression-free survival was longer in the patients receiving bevacizumab compared with the patients receiving placebo. And this is the Kaplan-Meier curve that you can see here is the patients that progress. At the beginning, you don't have any patient progress 100%, and then over time, you see the patients that start to progress. At the end, you can do a calculation, the hazard ratio, and you can say that it's significant. In this case, the p-value was very significant for saying that the PFS was better with the high dose of bevacizumab or Avastin. And then the question was, what happened with the data of each patient, if it really can fit the equation that I already showed to you. And these are 4 examples. This is a patient, individual patient. This is size of the tumor. This is days. This is a patient that only progressed with the treatment. This is a patient that has a beginning as a response, and then the tumor start to grow. And this is a patient with more follow-up that the tumor start to regress and then start to growth again, but it's more slowly. This month. And this is a patient with the really scattered data that don't fit this equation. This happens in more or less 100 patients in like 7 patients. I mean, almost more than 90% of the patients fit this equation. In this case, for example, you have only growth of the tumor, you can eliminate the decay part of the equation, and you left only with the growth part of the equation. And then they compare in these 2 arms the growth rate constant between the 2 arms. And you can see here that the high dose bevacizumab arm has more lower G or growth rate constant. This is exponential scale. I mean, lower means slower. And compared to the placebo arm, that means there's a statistical difference between the growth rate constant, but not really too much between the decay, how the tumors regress spontaneously in the case of placebo, or with Avastin in the case of the experimental arm. And then they did a correlation between the growth rate constant between placebo and high-dose Avastin or bevacizumab and overall survival. It was really a very good correlation, very significant, 0.6 and 0.6 for both arms. That means that this was the first experience using this equation that showed that really the growth rate constant is an excellent surrogate for FDA gold standard. But that time, now we know that PFS is a very common endpoint in clinical trials for FDA and can help us to discern effective therapies. What was the theoretical advantage of doing this and not the traditional endpoints by this time was that you can assess the growth rate constants before the RECIST criteria for progression, that means under treatment. And then I can show you another example, in this case, prostate cancer data. This was clinical trials done here at the NCI in castration-resistant prostate cancer patients using different combination of drugs. And again, in this case, instead of tumor measurements by CAT scan, were PSA values in the blood. That is a surrogate for tumor burden in the case of prostate cancer. And you can see here the same. Patients with, at the beginning of the treatment decrease the PSA, then start to grow. Here, a patient with just decrease during the protocol in the PSA, a patient with no really response and increase in the PSA, and also patients that you cannot fit their measurements in the equation. Again, in these 5 trials during 15 years was, I think, 9% of patients that cannot be fit in the equation. And this is the correlation for all of these trials between the growth rate and the overall survival. In this case is 0.72, very significant. The regression rate, again, the D of the part of the equation was not correlated with overall survival. And also there's another variables that were interesting to see. The initial PSA also correlates with overall survival, and that is normal because if you think a patient with a huge tumor burden will live less than a patient with just a small amount of tumor. And also the minimum, the nadir of the PSA will also correlated with the overall survival and the time to the minimum, the time to get this response. But if you see the correlation, the most higher correlation was with the growth rate constant, 0.72, if you compare with the other surrogates. And then this is the final analysis of all these trials over time at the NIH, using first for prostate cancer, thalidomide. This is an immunomodulatory anti-angiogenic agent. This is a combination of with chemotherapy, taxotere, docetaxel, hormonal treatment, and this is more aggressive treatment that combines Avastin, bevacizumab, docetaxel, prednisone, and then PSA Tricom. This is a vaccine. I mean, if you see, this is very interesting. If you see over time, the growth rate constant was, more or higher. That means more slow growth if you combine more drugs. But the last one, the PSA Tricom, this is the vaccine, was not really, they didn't follow this pattern and is the best of these treatments because it really prolongs overall survival. And what is this? It's just a hypothetical theory about this because this is immunotherapy. It's not really targeted agent or cytotoxic agent. This is the way how vaccines works is to trigger an immune response that it maybe is delayed to the fact that you are treating the tumor, in fact, under treatment. That means, for that reason, you have maybe a lower G in this case. And continue with kidney cancer just to see how we want to use kidney cancer as a model. We know there's still 14,000 Americans dying of kidney cancer every year. And if you compare this number to the year 2002, it's almost similar. And we have 7 or 8 more drugs approved by the FDA to treat kidney cancer. Really, the number of patients that are dying by this disease is the same. And this is, again, what are the drugs that we use for kidney cancer target is the VEGF receptor 1 and 2, mainly in the tumor cells and the endothelial cells. But mainly all the drugs target the same pathway. And then we start to analyze this. This was the registration trial for sunitinib and TKI, a small molecule, against an interferon. This was the immunotherapy that was approved for kidney cancer treatment before the TKIs. This was a big trial, 700 patients. This is again to show that this is sunitinib, a small molecule, blocks the ATP, the phosphorylation of ATP domain in the TKI receptor. And that is the way how it works. The difference to the VEGF agonist, the bevacizumab, is that it really is a dirty TKI inhibitor. This is a really idealistic diagram because we know that sunitinib blocks more than the VEGF receptor, blocks other things. And there's some subtle difference in these TKIs related to that. And this is the trials, the overall survival update of this trial. And this is the important numbers to remember for the next slides. This is the response rate for patients in sunitinib, 47% compared to 12% in interferon. And the median PFS, 11 months against 5 months in the interferon arm. And here you can see the Kaplan-Meier, the difference between sunitinib and interferon, almost 6 months, very similar to the difference to the overall survival. That means was clearly that was a better drug. And then Dr. Fojo and his group at that time, 4 years ago, got all the data from this trial, all the sheets of each patient measurements of the tumors, and they calculate the growth rate constants for sunitinib, and they look if it was correlated with the overall survival. And here again, in a log scale, you can see the more slow the tumor growth, more the patients live. And here in interferon, the same, but not so strong. You know, the correlation here was 0.37 and here was 0.66. And then we start to think one year ago when I arrived to the group, what happened with the second line in kidney cancer, how we can really know if we were using drugs very similar that target the same pathway, if we're really improving the treatment of these patients. I want to show you an example to reinforce the concept of the growth rate constant and the concept of the burden of the tumor. This is a patient with prostate cancer. They start treatment with a PSA of 10, then the treatment works, you know, clinical point of view works, the PSA drops to 2, the patient was keep on treatment, drops to 0.2, that is almost the nadir in this patient, the lowest value, then it started to growth again, 0.4. Everybody was happy with because he was using chemotherapy. He started with 10, but has now 0.4. And then 0.8, 1.6 is still very low compared with initial PSA, 3.2, 6.4, 12.8, and 25.6. And this time the patient was stopped chemotherapy, was put in another treatment. And this is an example for what? This is the plot of the PSA and the time, and you can see again the sigma plot and curve that show this initial decrease in the PSA values and then the increase in the PSA values over time. And if you plot this in a logarithmic scale, you can see this is a straight line. That means there's the difference really between this point and this point related to how the tumor growth is similar. Double in this case the PSA or the tumor, and in this case 12 to 25 is exactly the same. It's double the size. That means there's no acceleration in the process. The thing is that you have more tumor, more mass, but there's really no acceleration in any point. And just to keep in mind this concept, we try to look what happened in kidney cancer. It's just for a clinical point of view. This is the NCCN guidelines that clinicians will use mainly in the private practice or outside the research setting to choose our treatments for kidney cancer. Suppose that you see this to guide your decisions. And you can see here first line and second line, and you have 7 and 12 or 14 options because we don't know really what is best for our patients. And in all the options, you have a clinical trial. And we are talking about 10 years of targeted agents, and we don't have still one that is really clear, better than the other. And this is this trial, the AXIS trial, that was to really see what happened in second line in patients with kidney cancer being treated in first line. And just to show you in patients that received prior sunitinib, the difference between two agents was just 1.4 months in PFS, in progression-free survival. That's really not a huge difference for PFS. For an overall survival difference will be maybe a good one for approval, but was not too much in this case, in this subgroup analysis. And using this rationale, we think that what will happen if the patients theoretically will keep in sunitinib in first line after progression? How we will compare with this 4.8 months? And we analyzed the data again, the data from the sunitinib trial that I showed before. And this is an example of 24 patients randomly choose from these 300 patients. And I want to show you two things in this. Here you have many different patients with decrease in tumor size, partial responses, patients with no partial responses, just growth of the tumor, patients with the sigma typical curve, etc. And below are in blue dots is the growth rate constant. And you can see that in each case, more evaluations, you have a more consistent or more clear a growth rate constant that is really constant over time. There's no really fluctuations or accelerations. Here, for example, you see from here, or almost from the onset, you can see that the growth rate constant can be calculated that is similar. That means doesn't change. That means the growth of the tumor is being stable over time. And this is another way to look at this graph. This is all the patients, and we plot the G against the time, and we can in just 7 patients or 8 patients, we can see some acceleration in the G, in the growth rate constant, from 300. That means it's like 0.7% of the patient has some different behavior in the tumor. But really, in the majority of the cases, keeps constant. And really, we want to look what will happen if the patient keep on sunitinib until the second progression defined by clinicians by RECIST. And for doing that, we did this simple calculation. We used the natural logarithm 1.2 because it's the 20% increase for RECIST, and we divide this by G, the median G of all the population, considering all the patients, including the patients that got acceleration of the tumors. And this was 222 days. That means 7.3 months. This is a mathematical calculation, it's a modeling, it's not a trial, but it's hypothesis generated. And if you compare the second line after sunitinib in other trials, big trials, prospective trials, in theory, this compares very, very good, in theory. And for that reason, we want to push, I don't know if it will happen ever, but to do a trial comparing this strategy to keeping sunitinib versus switch to the other drug. And the other thing that I mentioned before, and that growth rate constant in this tumor and in this data set said to us is that they suggest that the resistance is intrinsic and not acquired because it's coming from the beginning at the same rate. And the other thing, well, again, for you remember the curves of the decay fraction, the sensitive tumor, the resistant tumor, and the resultant tumor that we see in the clinic in our measurements. And the other thing is interesting that you can really calculate that the growth rate without killing additional tumor can result in an apparent greater cell kill. That means that if you really change G, the constant for the growth, you can have an apparent better killing or PR or CR because if you double the G, the growth rate between the blue, green, and red and black lines, you can see the nadir for the response is different. But it's not because you are killing more cells, sensitive cells. It's just because the population of resistant cells is smaller or larger. And for that reason, the apparent results in the tumor that you kill is more. But it's not because of that. It's because really you have changed the velocity of the resistant population is growing. And this is some thinkings about this analysis. We collect data, and many times during the years we ignore it a little bit. But we have to look deeper in the data to get more information. We should think of a tumor as a dynamic entity composed of a drug-sensitive portion that is regressing and a drug-resistant fraction that is growing, as we looked before. We can measure the rate of growth of the resistant fraction, and this correlates with overall survival in at least 2 or 3 analyses that I present to you. In a given patient, the rate of growth of the tumor is as, if not more important than the absolute quantity of tumor. I put this in parentheses, if not more, because of course, if you have a patient with a huge amount of tumor in the body, independent of how the tumor is growing, that patient maybe will die because of the amount of the tumor in his lungs and his liver. But in general, this is a very important concept that maybe is very important, or maybe more important, how the tumor is growing. The rate of tumor growth when on therapy remains constant. It appears to have been constant from the beginning, suggesting we don't know exactly, intrinsic resistance, at least in a core of the tumor. Sometimes the best thing that we can do for our patients is keeping the treatment. And this is a really strong statement, but I think we have to at least assess or design clinical trials to assess this hypothesis. And in clinic, it happens a lot. As a clinician, many pulmonary oncologists and kidney cancer oncologists specialists keep patients in the TKI if the patient is well enough, is fit, and is clinically normal. Because the second option, we know, in the options that we have are not very good. And keep patients, for example, with TKIs in lung cancer for more than the progression is something that is done, but it's not really assessed in clinical trials, or we don't have good data for explaining that. A high response rate does not mean more tumor was killed by the therapy that achieved this. This is most often achieved by reducing the rate of growth of the tumor, as I showed in the last slide. By reducing the rate of growth, the sensitive tumor has more time to maximally regress and more evident in our evaluations. Most of our therapies are G therapies, meaning that they only reduce the rate of tumor growth and do not kill more cancer cells necessarily. And as a conclusion, using data collected as a part of a clinical trial and calculate growth rate constant and regression constant is very important, and we have to do it. We just need the sheets of the patient. Just in a couple of hours, you can do these calculations. The rate of growth correlates well with overall survival. Estimating these rates can give us ways to assess tumor growth and a better understanding of our therapies. And we can analyze a huge amount of data. It's like to have an experiment of 300 or 700 mice, and you can measure and get a lot of information, in this case, patients. And I think just PFS, OS, CR, PR, that is the manuscripts and the papers presented in journals is really just a part of the data you can get from all of these big trials. Estimating these rates can also give us insight into how we might alter therapies just by continuing, in this case, them for longer periods to achieve greater benefit. This is really work of Dr. Fojo. I'm just replacing him and just collaborating in some of his work. Wilfred Stein is the person who developed the formula, the equation, and works very close with Dr. Fojo. Julia is the statistician that they do all the plots and the calculations, and all the clinical team and all the collaborators of the urology and medical branch for all the data that I just showed to you. Thank you.
Very good job. Any other questions, comments, any discussion? So this is an approach that you were. Yeah. Turn your microphone on. Oh yeah. Okay. So this is an approach that you are using, and you would recommend be used.
Okay. In practice?
Yeah.
No, I think not yet. It's just modeling, mathematics. I try to understand from my clinical data, with the clinical data in a better way, how to assess eventually drug efficacy. But the thing, my goal with Dr. Fojo was to try to push for a trial, not big trial, but a small trial to try to prospectively evaluate this hypothesis and really see what happened when you compare sunitinib versus axitinib in second line. Because our projection is extremely good. Maybe it's not so good in fact when you will do the trial, but I don't think that will be interesting to see that, but at least will be non-inferior. And in many countries, like my country in South America, you don't have the options for second line or third line. And maybe the best way to do for your patients is try to keep, if the drug is working, and if the patient qualifies for many criteria, to keep in a drug that's really slowing the tumor growth. Yeah, but I will not recommend that for doing that in practice.
So you would think that this approach is applicable to any solid tumor, theoretically?
Theoretically, that is Dr. Fojo thinks that, I think we have to see the data. We have data for breast cancer, kidney, prostate cancer, multiple myeloma, that is a plasma cell dyscrasia, And now I will analyze data in lung cancer. I'm very interested in lung cancer because I think that is a very good model for TKIs and for keep-on treatment, and thymomas and thymic carcinomas. But there is a lot of data in many different tumors. I don't know if it will be the same. I don't know if it will be the same with immunotherapy, as I mentioned before. I think immunotherapy is a different animal related to treatment. I don't think that you can really calculate the growth, and it's a difference. The evaluation of immunotherapies is different, and I don't think you can apply that to immunotherapy.
I mean, when I see your data, I think about, maybe you'd comment about this, Michael, about diseases like melanoma, for example, where a primary melanoma is removed and they then go in, 5 years, 6 years later and look around for remnants of it and don't see any. And then 4 or 5 years after that, we have a brain metastases. Now, that's something different from latency of tumor growth alone, right?
Yeah.
I mean, presumably.
I think so. We have Dr. Stein developed another mathematical, put a more complex in the equation for adjuvant therapies. How really adjuvant therapy doesn't work very well, but.
So just a comment in terms of the mathematical modeling. I have a mathematician in my lab as well who's very impressed by these kinds of studies and points out that there's a second level of analysis. For one thing, in a heterogeneous tumor, you can't assume that every cell has the same level of resistance. So if you assume a variable level of resistance, you get a slightly different model. And the other thing is this exponential growth, which is interesting, but can't be always the case that the tumors are growing exponentially because they would slow down after a while. So you can introduce the idea that tumors will grow exponentially for a while and then as they get bigger will stop growing. It may be that what you're seeing in terms of exponential growth is not the exponential growth of a single metastases, but the exponential growth of the total tumor mass. Yeah,
exactly.
It's a slightly different situation in terms of how you would treat multiple metastases.
Exactly. Yeah.
There is this wonderful, interesting phenomenon in melanoma. There are about 50% of melanomas have BRAF mutations, which is another tyrosine kinase growth promoting pathway. And there are a number of different inhibitors for BRAF. And there are lots of examples of patients who are covered with melanomas. They get these BRAF inhibitors, and they totally go into remission. Tumor goes away. And then 3 months later, they come back, and if you look, for example, at skin metastases, every single one of the metastases has returned. Not just one or two or a few of them. So it's as if each of those tumors has within it the seeds of the resistance that will later develop, which is consistent with what you're saying. There are very few other mechanisms that can explain that phenomenon.
I will say that I agree with that. My point of view related to the models, I think you can do a model more accurate if you put like the gradient of resistance or density or other things. The only problem that I see with that is that the complexity of the formula will be less applicable in the routine practice. Because this formula is really, maybe it's a reductionist, it's like a physics formula to try to put in a very simple way. That maybe it's not so simple like this, but it's easy to do because it's just measurements. And in practice, you check other things like density or grade of resistance. We're still far away from that level of measurements.
Okay. Oh, hey, Bob, you have a question?
In some sense, I think this question is for you. You started off your talk saying that some people believe that there's intrinsic resistance, and you don't take that point of view. This last talk and just your last comment about the melanomas talk about the intrinsic resistance. So I'm wondering why you've come to that conclusion that it isn't intrinsic.
I'm not sure I said that. I think I said that some people believe that all resistance is intrinsic, and Mauricio and Tito, I think, I don't want to put words into their mouth. I'm sure you don't think all resistance is intrinsic. But a lot of the clinical behavior of tumors is as if all the resistance that we see is due to the initial heterogeneity of the tumor. And part of that is because we're treating tumors that are far advanced. There are examples in bacterial systems. So we're always interested in what's happening in antibiotic resistance in bacteria because same thing is happening. Things are becoming resistant. If you look, for example, at resistance to third-generation cephalosporins, these are very sophisticated drugs that have been developed to treat antibiotic-resistant bacteria. And you follow in time the development of resistance. There's an interesting phenomenon, which is that very early on what you see are the induction of sort of general mechanisms of resistance, pump systems and metabolizing systems, and so on, that are not very specific for the drugs, but allow some cells to survive long enough so that you start to see fixed mutations that confer resistance. So it could be that's happening in tumors as well, that there's an initial phase of survival, which depends on mechanisms that are more general, like transporters and so on, and followed by a more substantial resistance mechanism that actually allows the cells to grow and so on. If that's the case, and nobody has actually done a very careful evolutionary study in molecular terms of what's going on in a tumor, presumably we can do this in leukemias because you can get samples quite frequently. So one of the things we're trying to do is actually to look using the tools that we have. But that would change your approach because it would mean up front what you want to do is inhibit all the sort of general mechanisms like the transporters and the metabolism and so on, and hopefully reduce the potentially resistant population a little bit further. But I think it's going to be different for each tumor, and it may be from patient to patient, different mechanisms will be brought to bear. The other thing about the approach that Mauricio is describing is it's a very practical approach given the set of drugs we have now. It doesn't necessarily tell us how to develop drugs that will be more effective or combinations of drugs so that even those resistant cells can be treated.
Yeah.
Neither of you dwelled or mentioned, except you did in passing, Michael, animal models, transplanted tumors as a source for experimental study, both of mechanism and of drug responsiveness. Given the heterogeneity of the people either here or online, you might respond to that.
Okay, so there are the animal model which is popular now is called the tumor-derived xenograft. And that is a mouse generally without an immune system that will take grafts directly from tumors. So there's no intermediate phase of growing cells in tissue culture and worrying about this adaptation process that I described. And a lot of pharmaceutical companies are investing large amounts of money to use those kinds of models. So these are human tumors growing in immunodeficient mice. What I did mention is that we think in order to make real advances, we need an organ culture system ex vivo that we can use to study the growth of whole tumors outside of the animal. The downside is that it's easy to manipulate, it's easy to do imaging and molecular studies. The real downside is that there are components missing. The immune system is missing, nutrient flow is missing. There are a variety of things. We're not sure how important those are, but there's no perfect system at the moment. But we need better systems to study drug resistance because to get new drugs, we need to be able to manipulate the tumors in a way that we can't do in patients.
What do you think about the new MSG model? What do you think about the use of the NSG, the primary patient-derived xenografts?
Talking about the question is about these totally immune deficient mice that allow the growth of most human tumors. By the way, we also didn't say anything about cancer stem cells, which for the last few years have been a very popular explanation for the development of drug resistance. And there are a bunch of reasons, and Tito, I think, agrees that it's hard to explain the behavior of tumors based on stem cells. If there's a stem cell which can differentiate into all the different heterogeneous types of cells you see in a tumor, then, and you kill all the cells that aren't stem cells, then when that stem cell grows back into the tumor, it shouldn't be resistant. It should be sensitive, as was the original tumor. And in fact, the tumors that grow back are sometimes resistant. So I think that's an issue. I think there's a lot to be said, particularly for the orthotopic transplants, where you take a tumor, let's say a kidney tumor, and transplant it into the kidney capsule. Because that allows for the local environmental factors, the cell types and the trophic factors, to have an effect on the tumor, which we think are important. But whenever you do transplants, it's not going to be exactly the same as the tumor growing in its original environment. So it's not perfect, but maybe it's better. That remains to be proven, by the way. It hasn't been demonstrated.
Okay, well, thank you both very, very much for